See, there are mainly two different types of chromosomal abnormalities. One is numerical chromosomal aberration, and the other one is structural chromosomal aberration. I have talked about structural chromosomal aberrations in some of the presentations. In this video, I am going to talk about numerical chromosomal changes, which we can observe in certain members of a particular species. So, what are different types of numerical changes in the chromosome, and exactly what are the terms used for a specific type of chromosomal abnormality that we can learn by going through this video. So, this is a table in which you can have idea about the different types of numerical chromosomal changes, but you should know that such chromosomal changes could be seen in any diploid organism, whether that is a plant or animal. So there are thousands and thousands of species of animals and plants which are actually known to possess a specific number of chromosomes, because we very well know that every species of organism possesses a fixed number of chromosomes. So in case of numerical chromosomal changes, we find that some of the chromosomes are either missing or they will be extra added, or in case of plants even there could be change in complete set of the chromosomes. So accordingly we use the term euploidy. Euploidy actually refers to the variation in the chromosome number because of change in a complete set of the chromosome. And if just one or two chromosomes are extra added with the diploid number, or if one or two chromosomes are absent or missing in the diploid set, then that is referred as aneuploidy. So in case of euploidy, we have cases like haploidy, diploidy, polyploidy, and also auto and allopolyploids. I will discuss about these terms in detail. Likewise, in case of aneuploidy, we use the term hypoploidy when there is deficiency of one or two chromosomes in the diploid set, and we also use the term hyperploidy when there is addition of one or two chromosomes with the diploid number of chromosomes. So in this table we can see the different types of euploids. Haploidy. Haploidy means haploid set of chromosomes will be present in the individual. We know the case of honeybee drone, that is honeybee males are there. They are actually haploid. They have a single set of chromosomes. Actually, queen that lays unfertilized eggs, and from those eggs drones develop. So haploids are also there in certain populations of a species where single set of chromosomes are there. See, just for understanding, I am showing here A, B, C, D. Suppose these are four non-homologous chromosomes. It means the single set of chromosomes, they are being represented as A, B, C, D. Now, if there is a case of diploidy where two sets of chromosome will be there, then A, B, C, D will be represented twice. A will be present twice, B will be present twice, C is also coming two times, D is also coming two times. So here A, B, C, D, and we find that majority of plant and animal species are actually diploid. They have two sets of chromosome. Okay, every individual receives one set of chromosome from paternal side and other set from the maternal side. But in case of plants, mainly in case of plants, we find different types of polyploids also, and we can purposely create polyploidy also, mainly in plant populations, because plants could tolerate polyploidy. And if there are three sets of chromosome, then that is referred as triploidy. So A, B, C, D, which I am showing as non-homologous four chromosomes, then they will be present three times. Okay, so here A, B, C, D is coming three times, so it will be a case of triploidy, that is 3N. Likewise, in case of tetraploidy, which will be said as 3N, you will find that A, B, C, D, these four non-homologous chromosomes will be coming four times. So they are being represented four times in a particular cell. Okay, and we also know the case of autopolyploidy and allopolyploidy. In case of autopolyploidy, see, like in this case, this is actually a case of triploidy where A, B, C, D is coming three times, but we find that there is duplication of chromosome in the same species. Okay, so multiple of same genome means same genome is being, if it is being represented more than 2 times, then it is a case of autopolyploidy. And in case of allopolyploidy, multiple of different genome will be there. So here ABCD, ABCD, these are the chromosomes coming from one species, but PQRS, these 4 chromosomes are coming from a different species. And if these chromosomes then produce a individual, then that will be the case of allopolyploidy. Now the other case could be of aneuploidy, in which I just said that there could be case of hypoploidy or hyperploidy. In case of hypoploidy, just 1 or 2 chromosomes will be absent. Like in case of monosomy, we use the term monosomy, which is represented as 2n minus 1. So 1 chromosome is missing. See here, ABCD, if these 4 are non-homologous chromosome, then the other set will have just 1 chromosome absent. So ABC means D is lacking here. So this is a case of monosomy because 2n condition, complete 2n condition is not there, rather 1 chromosome is lacking, so minus 1. In case of double monosomy, see 2n minus 1 minus 1. This is represented as double monosomy. What we find that 2 non-homologous chromosomes are missing. So ABCD is one set, and in the other set will lack C and D. Suppose C and D, these 2 chromosomes are missing. So it will be a case of double monosomy. It means 2 non-homologous chromosomes are absent. In case of nullisomy, we find that a particular homologous chromosome will be totally absent. So here this is represented as 2n minus 2. Nullisomy means 2n minus 2. And ABC, it means D is completely missing. So that specific homologous chromosome D is totally absent because it is coming from maternal as well as paternal side, but from both sides it has not come. So the individual will be just ABC, and this is represented as 2n minus 2. So this is the case of nullisomy. In case of hyperploidy, as I said, that 1 or 2 chromosome will be extra than the diploid number. So this can be said as trisomy. Trisomy means one specific chromosome will be 3 in number. So this is written as 2n plus 1. So ABCD is one set, another ABCD set is there, but D is extra. So D is coming actually 3 times. That is why it is referred as trisomy. Then tetrasomy. It means a specific chromosome will be 4 in number. So this is represented as 2n plus 2. It means ABCD is one set, ABCD is another set, and then D is coming again. So we find D chromosome 4 times in this cell, and this will be referred as tetrasomy. In case of pentasomy, the formula will be 2n plus 3, and you can see here how chromosome complement will be there. ABCD will be one set, another ABCD will be a set, then D is coming again 3 times. So 3 plus these 2 means 5. That is why it is a case of pentasomy. So these are some of the terms which are used particularly in case of aneuploidy, and we find that majority of plants, they possess such kind of aneuploids. We find in case of even human beings, the cases of aneuploidy are there. This is one of the best examples to explain trisomy in fruit shape. You can see this particular happening in case of Datura stramonium. Okay, this is actually Jimson weed, and in Jimson weed we can see the fruit shape. So this one is the fruit, and a normal individual will have actually 12 pairs of chromosomes. So in case of Datura stramonium, 12 pairs of chromosomes are there, and the shape of fruit will be exactly looking like this if a normal set of chromosomes are there, means 12 pairs of chromosomes are there. Okay, so 12 pairs of chromosome, that forms the diploid number. Now we find that each pair means each chromosome may have its trisomy. So we can have 2n plus the first chromosome is extra 1, then second chromosome trisomy could be there, third chromosome trisomy could be there, So this way, all 12 chromosome trisomy is reported from this specific wheat, that is Jimson wheat. And you see the shape of the fruit, that is phenotype of the fruit, gets changed because of this trisomy of a specific chromosome. So this is a particular case in which one can explain that how phenotype of fruit shape gets altered because of trisomy.